Hi guys, Mrs. Grant here, and yesterday you guys had an opportunity to watch an ed puzzle over ATP and cellular respiration. And so we're going to break it down just a little bit more into more of its components and what cellular respiration really is. And you guys are going to be filling in what we would call a foldable. So pre-COVID, we would have this foldable. You guys would have to handwrite the notes and get images and trace them. And so I have made a digital version, and that's what you guys are going to be filling out today with the notes that I'm going to be covering on your screen. And so at any time, you guys can pause the video, take snapshots if you guys need to. The only thing I do ask is that you type out the answers. And I know that seems like, man, that's just a lot more work. But as far as retention and understanding, if you see the notes and you are typing them in, that helps you retain that information. So the only thing that I'm going to allow you to snip is going to be the image. But the only thing that I ask is that you guys type in those answers, and that just helps with notes just in general for retention. All right, so let's go ahead and get started. For your first tab, let's talk about the mitochondrion. Okay, so your whole lives you've heard mitochondria is the powerhouse of the cell. Yes, and we're going to explain why. So mitochondria are actually surrounded by a double membrane system, and so we have learned that in the endosymbiotic theory unit. And those layers consist of inner and outer mitochondrial membranes separated by an intermembrane space. And so the inner membrane forms these numerous folds, and we call those cristae, and you can see that here. They're really folded over, and so it also extends into the interior or the matrix of the organelle. And what this allows is for more contact and easier entering and releasing of molecules. So you can see here, it's really easy for those molecules to escape and to leave, and allowing ATP to cross in and cross out super easily. So I wanted to show you guys that, and that's going to answer your very first tab. So you guys are more than welcome to screenshot the image, but you guys do need to retype this information into your own words. So that's tab 1, mitochondrion. Tab 2, glycolysis. Ooh, okay, so glycolysis, as you guys watched the video yesterday, starts by using 2 ATP. I always think of it like you're going to a gaming arcade and you have to pay to play. And so in order to get any of those tickets as reward, you have to pay 2 ATP. So you have these ATP, and the thing is that by using those, it's necessary to help create this unstable molecule that forms pyruvate. And in the initial step, glucose is phosphorylated to form glucose 6 phosphate. So phosphorylated just means that it adds a phosphate to it, okay? And so after the second reaction, it rearranges the sugar to form fructose 6 phosphate. So fructose is a type of sugar. The third reaction adds a second phosphate group. And what that does is it forms this fructose 1,6 biphosphate, and it's observed by early researchers. And so what they're starting to find that in reactions like 1 through 5, the thing is that 2 ATP molecules are used up before any ATP is produced. So they're realizing the importance of that ATP. And so this part of glycolysis is referred to as the energy investment phase. And so the final product of glycolysis is pyruvate. And so really what you're doing is you have money to go to the arcade. Then you have to go and get their coins, and the coins is ATP. Then you use the ATP to play the games in order to get the tickets that you are going to be receiving. So that's kind of my analogy when it comes to glycolysis, is that you have to pay money to get tokens like ATP. Then you have to go through the process in order to get the prizes. So that's the easy way that I remember glycolysis. Tab 3, okay, citric acid cycle and the Krebs cycle. You, as far as what you need to know for the AP exam, they are not going to expect you to know all of these steps, but you need to know in general that once that glycolysis has started, it's going to enter the cycle. And what they're doing is you are adding and taking away these hydrogens. So you create NAD plus to NADH, and it just keeps going. And so this whole process is like the machine that's going to help you end up producing some more molecules for energy. So for your notes, as pyruvate is being processed, one of its carbons is oxidized to CO2, and 80 plus is reduced to NADH. The remaining 2 carbon acetyl units, so that is COCH3, reacts with a compound called coenzyme A. We also call that CoA. So we can see here in this image, you have acetyl-CoA, and then it drops two of those, okay? And then there's a 6-carbon molecule formed from acetyl-CoA and oxaloacetate. So the reason that this is called a citric acid citric acid cycle is because you are actually making citrate. And so what this is another way to get energy and electrons off of these molecules to then be used at a later state. And you can see that it's pretty cyclical. Okay, so that's citric acid cycle, and you will also hear it as Krebs cycle. And you're probably more going to hear Krebs cycle when it comes to testing. In college, I heard both. All right, so now we have our electrons. So we went through the Krebs cycle, and now we have some electrons that are now going to go into this intermembrane space, and we're going to use these electrons in order to produce these hydrogens and get these NADHs and NADH pluses to get some more energy. All right, so the electron transport chain. It's the third and final stage of aerobic cellular respiration. So just know that aerobic means that you need oxygen in order for it to work. Okay, so the third and final stage of aerobic cellular respiration, the electron transport chain accounts for most of the ATP. So this is where you're going to get the most bang for your buck. Okay, so it's kind of like you have played all the games and now you're playing the best game that's going to get you the best amount of ATP in terms of investment. So the stage 3 transfers the energy from NADH and FADH2 to make ATP. So during electron transport, energy is used to pump hydrogen ions across the mitochondrial inner membrane from the matrix into the intermembrane space. So a chemiosmotic gradient causes the hydrogen ions to flow back across the mitochondrial membrane into the matrix. Although ATP synthase, okay, and so let's see how I want to say that. Through ATP synthase, it's going to produce this ATP. So by going back and forth and going across those gradients, you're actually generating this energy molecule. And so when the ATP from glycolysis and the Krebs cycle are added, you get a total of 38 ATP as a result from aerobic respiration and of one molecule of glucose. So glucose is sugar. And so sugar is broken down, and so you change it, that's what glycolysis is, and then you change it to pyruvate. Pyruvate is then used in the Krebs cycle, and then in the Krebs cycle, it's generating more energy that then goes through this mitochondrial membrane that's in the mitochondria. And so by going across this gradient, you're actually generating these energy molecules and creating more ATP. And so that's how we get energy. So remember, glucose is sugar. So anytime that you're eating one molecule of sugar, which obviously like one just little cube of sugar is a ton of energy, your body uses that to break down and gives you the quick energy. Okay, so let's talk about fermentation. All right, so there's going to be two types. There is lactic acid fermentation and alcoholic fermentation. You guys are probably more familiar with what we call lactic acid fermentation. If you have ever been sore, what'll happen is you are burning energy that you are not getting nutrients from. So let's say you're running a marathon, you're on a cross country meet. So you're running. So what you're doing is you probably intook a bunch of glucose, like that can be in the form of simple carbohydrates. So that would be anything like a sugar energy drink. And so what you're doing is you're giving your body that pyruvic acid. And what you're doing is, in this case, it's taking that NADH, turning it to ND plus, and what it's doing is it's going to send it back to glycolysis. Now, that's awesome. You're getting energy. However, as a product of that, you build up what we call lactic acid. And what lactic acid is this molecule that sits near your muscles that does not allow your muscles to function as well as they normally do, creating this soreness. So it's really those muscles are not able to perform at their best and you're getting sore. And notice how it doesn't happen until after you have burned your energy. So typically you do not get sore until after you work out. And as an athlete, you typically do not feel it until about 3 days after that major pull from your body's energy. And so that's why athletes, let's say you haven't worked out in a while, on day 3, you actually are the most sore because that's the most collection of lactic acid. So alcoholic fermentation, hopefully you don't have too much experience from this, but that is actually how we create wine and beer as well. So what'll happen is that you have this pyruvic acid, and what it's going to do is it's going to CO2, which gives it the carbonation, and you're going to produce ethyl, and that is the alcohol part of the alcoholic fermentation. And so fermentation is ultimately the breaking down of glucose and creating the byproducts. And in humans or in any animal, you create lactic acid, but for other, let's say alcoholic fermentation would be like death or decaying materials. In the case of grapes, that's where you get ethyl is produced. So ethyl alcohol. All right, so here's some key points to take away from this. Fermentation happens in anaerobic conditions, in other words, without oxygen. Fermentation begins with glycolysis, which breaks down glucose into 2 pyruvate molecules and produces 2 ATP and 2 NADH. So what this is saying is if you do not have enough oxygen in your body, so if you're working out and you are not giving your body enough oxygen, your body is going to go through fermentation. And so fermentation allows glucose to be continuously broken down to make ATP due to the recycling of the NADH to NAD+. So without fermentation, the electron carrier would be full of electrons. The entire process would back up and no ATP would be produced. So yes, you need fermentation. But if you do not give your body enough oxygen, then it gets to the point where it builds up. All right, lactic acid, in other words, lactate. Fermentation occurs in some strains of bacteria and in skeletal muscle and produces lactic acid. Alcoholic fermentation occurs in yeast and produces ethanol and carbon dioxide. Fermentation only produces 2 ATP per glucose molecule through glycolysis, which is much less than ATP with regular like cellular respiration. So ultimately, cellular respiration, and you need oxygen in order for this to work. All right, so you guys can take notes on that. And that is going to be concluding it. So, yeah, if you guys need any help with those notes, or if my little screen down here is taking too much up, you should be able to see those notes. But yeah, at this time, you guys can go back into those guided notes and be filling them in. So you need an image. It'll tell you whether to put an image on the upper or lower, what we would call flap. And then the second part, you guys are adding the notes, which is kind of the lecture that I've been given today. The lecture today was specifically for the foldable, so it should answer all of those questions. Let me know if you guys need any help. I'm still in professional development Tuesday, and I'll be in professional development Wednesday as well. So keep letting me know if you guys have any questions. Thanks, guys.